Hi, everyone. Welcome to the second episode of the Environmental Biotechnology Innovation Centre podcast, in Biotech. My name is Nicola Heaven. I'm EBIC's Comms and Knowledge Mobilisation Officer. As a UKRI Engineering Biology Mission Hub, the team at EBIC are made up of a fantastic team hailing from over our 10 partner universities, bringing a wealth of knowledge and experience in some fascinating fields. Our topic for this episode of the podcast is around barcodes, computer science, and engineering biology. Joining me here today are Professor Natalio Krasnogor of EBIC and our partner university, Newcastle. And we also have research assistant Yasir Akbar from Natalio's team at Newcastle. So welcome to both of you. To start us off, Natalio, would you like to introduce yourself and a bit about your background?
Yes, sure. Well, first of all, thank you very much, Nicola, for giving us the opportunity to talk today in the podcast. This is exciting. Let's see how it goes. So, I'm a professor of computing and synthetic biology at Newcastle University, and I have a Royal Academy of Engineering chair in emerging technologies, where I look at DNA data storage. And I'll tell you a little bit what this has to do with what we are talking today, which is barcoding. You were asking about my background. I did my undergraduates in Argentina a long time ago, essentially informatics and software engineering. Then I came to the UK to do PhD, and I did PhD in machine intelligence for structural bioinformatics, in particular protein structure prediction and protein structure comparison. And then I took an academic post in Nottingham University, and was still mostly in silico computational, but then after a few years set up a small lab, experimental laboratory in Nottingham. Then the research group outgrew, and we had a possibility here in Newcastle to expand that. And now we have a nice research group which works at the interface of computing and the life sciences. And we have a very nice resource wet lab where we do engineering biology, synthetic biology, as well as RNA and DNA nanotechnology.
Perfect. Thank you so much. Yasir, thank you as well for joining us today. It's really great to have you on with us. Could you introduce yourself and tell us a bit about what your work is at the moment?
First of all, thank you, Nicola, for invitation to the podcast. My name is Yasir Akbar, and I am research assistant of bioinformatics and biosecurity at Newcastle University. I am the part of the research group called Interdisciplinary Computing and Complex Biosystems. I am working in the cyber project and work closely in the collaboration with GitLife Biotech and of course EBIC. So my project Cyber is mainly focused on the reprogramming of cyanobacteria for environmental restoration, like pollutant degradation and oxygen production, etc. If I talk about my background, so my basic degree is in microbiology, and my further studies is in bioinformatics. So that's why I work in the interface of microbial bioinformatics. I'm more focused towards the genome of the bacteria and cyanobacteria.
Thank you very much. I think it's really fascinating to me how both of you with your work, you're on the interface really of computing science, natural sciences. So I know I've heard a lot. Many people think of biology as being maybe more like messy, and computing as more precise. How do you reconcile those differences if you're doing experiments, building models and things like that? How does that work?
Yeah, that's a very good question, Nicole. I think biology is messy and computing is precise, but biology is not as messy as people think it is, and computing is not as precise as people think.
Okay.
So I would say that there is a lot of things in common between biology and computing. If you take the genome, for example, you can consider the genome as a piece of software, which is a legacy software, which is billions of years old of legacy, and we need to deal with that software. That software has instructions to manufacture the hardware that runs inside a cell, the hardware that is a cell. So the question is how you deal with legacy software if you want to make them do new stuff. And this is what engineering biology really is about, and what synthetic biology is about understanding that legacy software and putting to work doing new stuff. So that's how, in a very broad, abstract way, biology is similar to computing. But there's the other way around as well. The most complex artifacts that our civilization creates are not jumbo jets or spaceships or cars or mobile phones. It's software. And it is so complex that there are emergent properties in this software called bugs. These things that really annoys every user when the computer crashes or you cannot connect when it's supposed to connect, and so on and so forth. These are emergent properties, and they are emergent because you cannot predict them upfront. You cannot prove everything about your software that you would like to prove or demonstrate theoretically that it will work perfectly. And this is where software starts to approach living systems. And if you look at the evolution of software, you can even when you do data analytic on how the evolution of software actually it looks pretty much like the evolution of genomes with how you call this phylogenetic trees and mutations and things that get frozen, accidental features of software that get frozen in time and then get reused and repurposed, etc. So there are lots of commonality between how biology works and how software works. But I think that ultimately, in my mind, biology is digital. I think it is programmable. It's just that it's a different kind of programmability than what one thinks in terms of programmability, like normal programming languages. So the opportunity there is for both the biologists and the computer scientists to work together to find out how you can program biology in a better way, which is not exactly computer science and it's not exactly what I would call old-fashioned way of looking at biology. And this is where the real interesting challenges and opportunity, I think, lies.
That's perfect. That makes sense, yes. Yasir, is there anything that you wanted to add to that or expand on? Or
So I think like similarly, biology can be messy and unpredictable, while computing can be used to get insight from those biological data. Like we can use the tool to analyze the biological data and make sense of those complex data. It helps us to find a pattern and make prediction, and extract something from that biological data. For example, if we talk about the NGS data, it's produce a million of reads. But how to deal with this million of reads? Obviously, the computational tools. If we don't have computational tools, we cannot assemble the genome, we cannot check the quality, we cannot annotate the genome. But the thing is, they work together. For example, the sequencing machines and the reads. We analyze those sequences, we annotate. Then we use computational tools to predict something, or we prioritize candidates. For example, if I talk about my research, the nucleosides, we have thousands of genes, but we use computational tools to prioritize the candidate genes. But again, we need experiment to validate. So this is like a cycle. Experiment, computer, experiment, computer. It's like goes on an iteration. And if we see with the time, as the software getting advanced, it become more and more accurate. If we talk about the AI, so previously, some years back, the AI was not that much accurate, but today it's performing brilliant. So this is the case in the case of computer science. So when we get data, we actually use those biology to optimize our software, to optimize the parameters so it can perform better and better.
perfect, thank you. So, do you think what have you found the biggest hurdle in sort of helping maybe computer scientists to think biologically, or natural scientists to think a bit more computationally, or was it more like my perspective that obviously I didn't realize how similar they can be? What do you think there? Are there hurdles to come across?
So first of all, biggest hurdle is definitely how the two group thinks. and the mindset, which is because of the years of study. If one person study from the beginning the computer, and the one person studying, he is just limited to the laptop and the screen, and the one person is studying the biology and doing experiments, going to field, going to lab and benchmark, obviously it will create a different mindset. So if we talk about the computational things, so the computer scientist actually depends on the values and the results they get from the analysis. And the important thing in the biological analysis is the linking of those computational results to biology. For example, if you talk about the normally in the statistic, the p-value. So normally we have 0.05 considered as significant, but in biology we sometimes need low p-values like 0.01. So it is known to, if you know about those interface, the biology and then computer science. So you have to link your results to the biology. And if I talk about the natural scientists, they usually deal with the experiment, the messy data, they work on the bench. So initially it's hard for them to use to the dry lab things.
Mm.
And I think now in this era, it's not the case like this, but if they are early career in their career, so they will definitely learning to trust the computational tools and models, because in biology matters to them most than values and the results they are getting, and they think whether the results has biological meaning or not. Or sometimes they feel like computer oversimplify the biology, or the coding is too technical. Like for example, they are doing experimental if they are not getting results, and we perform the analysis and we told them yes we performed it, so they think that how we are not getting results and you are telling us that we did it. So they think we may oversimplify those things. But in conclusion, we can say that like if they both work together and respect their uh, ideas, and so we can combine those fields and those two scientists, whether for computational biology or experimental or natural scientists. So if they work together, they will perform better and better for good future in science.
Perfect. That sounds good. Did you want to expand on anything or add anything now? Are you happy?
No, I think what Yasir is very good. Just a couple of observations, perhaps um, coming from we have masters in bioinformatics, synthetic biology, these kind of things. And some of these masters bring together people from computing that wants to get into biology, and people from biology that wants to get into computing. And I think, notwithstanding that there are very different kinds of computer scientists, because computer science is a big discipline with lots of two disciplines, and there are lots of different kinds of biologists. It's a big discipline with lots of subdisciplines uh, inside biology. There are some things that you could say perhaps apply almost across the board, which is that in general in biology, you try to understand the problem. You have a problem question, a research question you have, and you try to understand that that question to understand a mechanism, let's say, or a process. In computing, there is a lot of effort on a synthesis, on the creation of new stuff. So yes, of course, you want to understand things like complexity and algorithm design and all of this, but actually it's because you want to build something. You want to build an algorithm that works faster or that solves a problem. And and these are very different way of looking at the world, I would say. So and this when you need to get people to shift or work cross disciplinary brings a tension or friction. I guess for a computer science person, one of the difficulties in getting more into biology is that there is a lot of fragmentation in biology. There are millions of different databases, different data sets all over the place. There are no clear standards. The jargon is horrendous. The microbiology would name things one way, the plant biology is another, the animal biology is another, and sometimes they are talking about the same thing or better thing. So and this makes it very, very difficult for somebody that comes from a computer science degree to say, okay, where do I start with all? I start with all of this. on the other hand, for the biologists, since they are trained, they are quite trained in a quite reductionist way. So they perhaps don't get to see that there is those fragmentation because they have been looking very narrowly about molecular action on a molecular substrate, and this is what they get to see. But what they mean by that is to learn to think abstractly and more in a divide and conquer way, like a computer scientist can do from probably after the first year of computer science. You can take big problems and start to divide smaller problems. So, so I think these are very different way of training people. There is a sizable shift now in biology because it's moving towards synthetic biology and engineering, which is about building stuff. And then you start thinking with this system level engineering where you say, this is my system, these are my subsystems. I don't need to understand everything. I just need to understand some of the interface with this, and what happens inside maybe is not important. So I think this is a dialectic that keeps going on. It started with bioinformatics many 20, 30, 40 years. But now is being pushed much faster because of engineering biology and synthetic biology.
No, that makes perfect sense. So would you say, is there a computational tool that you think all biologists should learn to use, or maybe something computer scientists should take from biologists? Is there something, what would be your main recommendation, do you think?
So, okay, I'll just start with it. I could follow from the periodical, then I'll choose. So Yasir may add something perhaps more concrete. Uh, I actually don't think it's about tools.
Yeah.
Because tools become obsolete very, very quickly. So I think it's about principles. And there are fundamental principles from computer science and certain fundamental principles from biology that are mutually useful and cross-feeding. So I will tell you two principles from biology first that computer scientists have onboarded to very good effect. Darwinian evolution. Evolution by natural selection. Computer scientists took those principles, 30 years ago and developed whole fields of machine intelligence and optimizations around Darwinian evolution, which now is applied everywhere. People don't even pay attention anymore about this, but it's being used everywhere. And the other one, of course, is neural networks. All this the hype and wuhaha about artificial intelligence is about neural networks. These come from biology. These are principles that were learned from biology and then operationalized and scaled up and formalized in computer science. So computer science, in that sense, will learn principles from anywhere. There is a complete discipline, subdiscipline within computer science called nature-inspired computation. I'm trying to borrow principles to automate and orchestrate those principles into practical tools. The kind of things that I think principles that biologists would do could benefit from is, again, what I was talking about before, about understanding abstract reasoning, system-to-system modeling, divide and conquering, learning to do knowledge formalization, whether you formalize the biological knowledge you had in your head in rules or in computer programs or in mathematical models. It doesn't matter. Any kind of formalization is better than writing a narrative in natural language, because natural language is messy and leaves lots of in the middle and is non-reproducible. So I think there are lots of things that could be learned from computer science from the point of view of principles, right? And a specific tool that today is fashionable, tomorrow is.
Yeah, very good point, to be fair, isn't it? Everything changes so fast. Yeah. Yasir, is there anything that you...
Yeah, so I will answer according to my experience when I spend time with my friends. So those friends which are actually working on the experimental sides.
Okay.
So I will answer for my experience what kind of question they ask from me. So most of the time, my microbiologist friends, they have problem with statistical analysis, like correlation, chi-square, t-test, and these kind of things. So if I would tell if the biologists learn those statistical analysis skills. If they have the skill of the statistical analysis, and nowadays it is very easy to perform this kind of analysis because even the Excel now is very powerful and it has a certain kind of statistical analysis. So first of all, I will recommend those to learn the stats. And the next after this, they can add certain skill, especially for the microbiology. I will recommend the metagenomic analysis. And there are certain tools available, like Galaxy and other GUI-based tools, which will really make the analysis easy for the biologist. And other skill set, they can then further increase their skill set. So it's very important now, even for a biologist, to learn some basic computational skill, even like WGS. Maybe not that much at the advanced level, but uh, that much that they know what is the reads, what kind of sequences I am getting from the machine. So they know about this. So
That's great. Thank you. That makes a lot of sense as well. So going from sort of between biology and computers, is there a way that we can make the field more accessible or legible, perhaps, to non-experts? Because obviously, it's dealing with both. You're looking at code, you're looking at cells, you're looking at complicated topics separately, let alone both together. Is what would you say is the best way for us to kind of communicate this, as I say, to non-experts, to the public?
I think this is a really, really hard, and I have so many conflicting things going in my
Yeah.
head. What thinking about this? Uh, I think one thing that I would love is that we stop mystifying biology, but also in computer science things like AI. There is all mystification of AI, and there is certain mystification of biology. I think both are wonderful, but ultimately, in neither there is magic. There is no magic in computer science. There is no magic in biology. There is only physics. There is only chemistry, and there is only information process. The beauty for me of the natural world is not that it's some sort of sacred realm of some way, but rather that despite of not being some divine realm, is knowable. It's something that we can get to know, even in all its complexity and in all its beauty. And because of that, it's worth preserving and nurturing it and protecting it and trying to understand. But when people start to attach moral dimensions to AI or to biology, this is where any kind of discussion becomes really, really difficult
Yeah.
because there is no compromising on morals. This person has one moral view on
Yep.
an aspect of the world. This person has a different moral view on the aspect of the world, and there is no negotiation there. But if we people to understand how the world works, and I'm thinking things like vaccination, for example.
Yeah.
You can see what's going on in America right now, and probably will happen in the UK soon if certain situations take place politically. Uh, well, at that point, the logical discourse, the rational discourse goes out the window, and that is a real, real shame, I think. Uh, and I'm very, very worried about this. So I know what is the answer, but I know that moralizing it is not. I think it needs to be from a point of understanding the biology is hard. It's very, very hard. It's very, very complicated. We don't know everything, but oh boy, we know much more than we used to know even 5 years ago, let alone 100 years ago or years ago. So I think all the bashing of experts needs to stop. And I think we need to be more vocal saying, we know what we are talking. And when we don't know what we are talking, we need to say, we don't know what we are talking. And no, we do not know it. We don't know everything. Of course, there are things that we don't know. But there are other things that we actually do know. And this, we need to be clear about this. At least that's
That
my
that makes perfect sense, and that's I think that when we're open to say we do know this, we don't know this, you kind of hopefully going to be able to build more trust as well, aren't you? If you're open and you're honest, I think that's definitely a good starting point, at least, isn't it? So
I think the impression we need to stop giving people is that science right now knows everything. That's is never whenever is now, whether now is today or in 100 years time, or in 1,000 years time, there will always be things that science does not know.
Yeah.
That is the nature of science. Pretending otherwise is self-defeating, I think.
No, absolutely. That sounds good. What do you think, Yasir, if you've got any extra points you wanted to add in?
So I will say that if we talk about the accessibility, so we can, like, you can provide accessibility in a way, like, for example, if we explain certain ideas in a simple language and use example, just like, yeah, like just to educate them that how these things are actually working. So if we, on the other side, nowadays, if we talk about the social media,
Mm.
so because of social media now, people know a lot about DNA, the genome. Even the people with the completely to the literature side fields like English or other languages, they also know about the DNA genome and even the genetically modified organism GMOs. And they know that we can buy GMO fruits or vegetables inside the market. So I think in terms of accessibility, we can like explain the things to them in the simplicity. Just they should educate about this, like what we are doing and what we can achieve from this. So if in the future, if you want to explore those fields, so they should have to come with a proper way, the proper channel, not to just give direct accessibility. Because in certain cases, the genomic data can also be as sensitive. So giving it to the people which are not expert can cause harm. Also, there is a chance. For example, the research area is separate, but if we talk about specifically about the clinical side where we deal with the data, genomic data of the patients. So there we will be very careful. So yes, we
Yeah.
can provide a little bit of accessibility to educate them that how the genomes are working, how the code and cells are working together.
Great, thank you. So one thing that I'm really interested to talk to you both about today is actually your work with barcoding systems. Can you explain a bit about the barcoding in this context? Because obviously, I'm sure everyone's all familiar with barcodes. You do your weekly shop, there's barcodes everywhere. But what have they got to do with engineering biology and the sort of the field as a whole? What are they?
Sure. Um, so yeah, I think the point is exactly what you say. Barcodes are everywhere in the supply chain. Uh, any product you buy in the supermarket or in B&Q or in IKEA will have a barcode. And why there are barcodes in the supply chain? For a number of reasons. First of all, to the shop concern, let's say the IKEA or the B&Qs or co-ops or whatever, it allows them to keep a clear inventory of what is in stock, what is not in stock. They maintain track price variations, number of amount that is sold, etc., etc. But the barcode also tells you what the product is, unequivocally, who built it, where it come from, when was it built, what was the batch number, what parts are inside, because inside, let's say, a computer, there are lots of parts, and inside each part there is a barcode. So from one barcode, you have lots of barcodes inside. So essentially, it allows, it's like oil in a very complex machinery, which is a supply chain, that makes the machinery go operate smoothly. For example, sometimes you will go to the pharmacy or to the supermarket and you will see notices saying, we are withdrawing from the recalling such and such product, barcode such and such. So if something went wrong, for example, the product is faulty or there was a contamination or whatever, you can recall a specific batch of products and you will, and the supermarket or whatever, or any shop will know when this batch was sold, in which period of time was sold, in which branch it was sold, etc. So you can record the product, you can minimize harm, in the supply chain to ultimate end users. So there are lots of reasons why barcodes have become so popular. There is no uh, uh, agreed barcoding system for engineering cell lines. I want to make a distinction. There are barcodes are used in the life sciences, but usually are used for something quite different than what we are talking here. They are not tracking the supply chain of genetically engineered or recombined cell lines or plasmid and these kind of things. And the reason you want ideally to have a simple system of inserting barcodes, reading barcodes, tracking them is for the same reason that we said before. Because whomever receives a cell sample that has certain barcodes, then you will be able to verify very quickly, do I have what I order? Is this coming from the right person, the right laboratory? Hold on, this cell line comes from this laboratory, but I don't see any registration that they have followed the Nagoya protocol for importing cells from another country from wherever the sample was taken. So there are things like biodiversity preservation, access and benefit sharing that could be better tracked if there were some barcodes used routinely. Unfortunately, that's not the case. Another example of if the barcode examples I gave are not convincing, maybe this one will convince. Imagine if we were driving in our roads and cars would not have registration plate numbers. Okay, so what does it mean if we didn't have registration numbers for the cars? Well, what it means is that if you wanted to buy or sell a car, you wouldn't really be sure you are buying a legitimate car from the legitimate owner. It might have come from somebody else. If your car is stolen, how do you know? It's more difficult to track because they don't have registration. If a car or a bus is involved in an accident, how would you determine who was the owner of the car if they just left, for example? But because there are registration numbers in the cars, then the insurance kicks in, and it's easier to buy and sell cars, and it's more difficult to sell a stolen car and so on and so forth. So there are lots of reasons why having a proper tracing and tracking system for engineering cell lines, we think is the right time to introduce it into the discipline.
Okay, thank you. So going off the back of that, so what is the process of designing and integrating a barcode? What does it actually look like to you guys in the lab? Can you just describe that for me? Because I can't picture it personally.
Yes. So in a sense, it's reasonably simple. It's just a unique piece. It's a unique sequence, which in the digital world is a zeros and one sequence, or rather hexadecimal sequence that gets converted into DNA. And this piece of DNA contains your barcode. It's like if it was your car registration number or plate number, I think some people call it. And you can insert that number inside the cell line. So it's essentially a piece of DNA that you can insert into an organism. The specific way of how you can generate a given barcode, probably we don't need to get into it. But it's not. There are relatively simple ways of doing that. For example, you could just generate a complete random sequence and just keep a track, a log, say, well, this random sequence belongs to this lab, to this species, whatever, and you insert that in the organism. I guess the part that perhaps is more involved is the kind of thing that Yasir is looking at, which is trying to say, okay, but where do we put it? So it doesn't affect the rest of the organism. So I will not say anything about this in case Yasir wants to add a little bit. But after you know where you want to insert it and you know what the sequence is, you just do quite standard recombinating that any laboratory that does synthetic biology or engineering biology, they do day in, day out, and you can insert that barcode in the cell. So it's not rocket science at all.
Brilliant. Thank you.
Can I discuss some computational approaches for designing, like in general,
Yeah.
how the barcodes?
Yeah.
So I will say barcode is discussed by Netter, random DNA sequences, but there are two important things to be considered. One is length of the barcode and degeneracy for the barcode, and the other thing is the complexity. The general structure for the barcodes can be like N3WS, where N is actually a degenerate nucleotide. It may be A, G, C, or T.
Okay.
And S and W is a partial degenerate, where W is for weak base like A or T, which actually form two hydrogen bonds, and S is for strong base, which actually form three hydrogen bonds like G and C. The length of barcode is selected on the base of the amount of information we need. That is how many unique barcodes we want to, and it depends on the number of strands. And the second thing is composition, like GC content and the complexity. We try to avoid low complexity, like long similar sequence, for example, A or G, which actually create problem for even synthesis. And the next thing is when we generate computationally, and we try to synthesize it and then insertion to the organism.
Perfect. Thank you. That sounds great. So at EBIC, we know that traceability of engineered microbial strains is key to public trust and also things like regulatory compliance. So how does barcoding fit into EBIC's strategy for monitoring organisms?
Yeah. So what we are trying to do in EBIC and the other project that Yasir was mentioning, cyber, is essentially we're trying to advance engineering biology tools so we can start solving very important environmental problems we had with engineering biology tools. But right now, it's not really possible to release into the environment genetically modified organisms, so even if they are even if they are not GMO and they are precisely edited organisms, still there will be lots of hurdles, regulatory hurdles to go through. But one of the, I suppose one of the cross-cutting or complementary components of having a healthy discipline of engineering biology is making sure that when we engineer an organism to do X, there is a way to make sure that people can have access to the information that led to the creation, the engineering of that organisms. So when you have barcodes, you can do tracking and tracing, which essentially enables you to do forensics and attribution biosecurity. Because if an organism, if you have a sample of the organism, you can obtain the barcode, and without barcode, you can go to a digital record and see how that organism was engineered. Not only it will tell you, okay, this organism is this organism and was engineered in such and such lab, but also will show you or should show you the process how the organism was engineered. So you can have more transparency, you can have more trust that what is being used for, I don't know, metal recovery, let's say,
Yeah.
or some other environmentally relevant application, has been scrutinized and is continuously available for scrutiny. So it's not that some office in some lab is saying, okay, here is a solution, and they drop the solution onto the society.
Yeah.
Beyond that. So yes, we know what we are doing. And because we know what we are doing, we make it transparent. We associate a barcode that allows us to go and from this barcode see a register of how this process of engineering took place, how the organism can be tested, and the what condition it is safe to use, and the what condition is not safe to use, etc. Now, this doesn't mean that the entire public will have access necessarily to all this information, which in some cases could be proprietary from some company, for example.
Yeah.
But it does mean that the regulator, which is whom the public should trust, should have access to that information. So it's always about making the infrastructure, the general bioinformatics infrastructure, available, so the right kind of process can be built on top of that. This doesn't right now. So we are trying to see, we don't have exactly a solution how it will all work out, but these are the kind of principles we want to do. We want to make the tracking and tracing more transparent because it enables forensics and attribution of where these organisms come from. We want the process of engineering itself to be more transparent using things like version control, like you have in software engineering, for example. So multiple people can work on the same project without stepping on the toes, and it's clear who is doing what and for what reason, what's the intention behind the modifications to the algorithm. And when you combine both, you have this enhanced trustworthiness, transparency that I think should be desirable anyways for this kind of disciplines.
So building on what you were saying then, Nat, so if you were approaching, say, conversations with people who maybe are a bit skeptical about synthetic or engineered biology, have you found that being able to explain about barcoding, do you think it does reassure people in terms of sort of accountability, traceability, things like that? Do you think it's kind of a good thing to be able to have?
In my experience, yes. And I had an example. Last week, I have somebody visiting from Argentina. This lady is involved in what is called a community-supported agriculture, and they are and essentially there is a farm, and there is a system of the local community supporting the farmers that run the farm and essentially paying them forward for the crops that they will produce. So the farm doesn't need to go and sell their products at ridiculously low prices, and they are not outcompeted by very big corporations. And because they have their livelihood secure, they can invest in more nature-friendly ways of dealing with replacing chemical insecticides and this kind of thing. So it's good for nature, it's good for the farmers, it's good for the local community. They eat locally sourced food. So they were talking about what we do in EBIC, and I explained, and the first comment that came, we said, "Oh, but so you're modifying microbes?" Yes, we are. So you're playing God. And this is where, and which is always the first initial reaction that have, oh, you're playing God. So this is where we started explaining, well, actually, you don't need to be God to be able to do that. Actually, you can be just a humble scientist in a lab, and we understand how this works. We have been doing this kind of thing for many, many years. And then the follow-up question was, okay, but if something goes wrong, how do you know if you were to release one of these in the environment, how would you know that they come from your lab? And that was exactly what barcodes are. And I said, well, because first of all, currently we are not releasing anything to the environment, just to make it clear. But if we were to release it, it will always have a barcode. And it will have a barcode the same as you have a barcode for your yogurts or your pizza. You have a barcode, and you will be able to recall your organism because you will know where the problem started, you will know where the origin lab was, and then you will know with whom to speak to try to solve the problem. You will not need to be doing a very complicated detective story to try to find out how to solve it. You will know straight away what needs to be done. And that completely shifted the conversation because people are familiar with the concept of barcodes. And they can see, oh yeah, I can imagine how that could work, and well, the conversation took them in another direction.
Yeah.
But that's one example how I think it does help. It's not the solution to everything, of course. There is always this issue about the playing God, and this is why I was talking earlier about this demythifying biology. But it's a step in the right direction, I would say.
Brilliant. Thank you. So, if any, are there sort of any limitations of standard barcoding approaches currently?
Well, yes, there is a big one. That
Yeah.
there are no standards.
Ah,
We do not have any standards for genomically inserted or precision edited barcodes, and that's of course is a bigger limitation. So we, as part of EBIC and in collaboration also with GitLife, Biotech and the British Standard Institution, we ran last year a scoping workshop to look at this issue of the standard. It was really nice because there were, I don't know, 30, 40 people, I don't know how many, lots of people, only a few academics,
mhm.
lots of representative of different industries. There were colleagues, experts on metrology, and of course experts on standards. And it was not difficult to agree a scope for a standard, just a minimum scope for a standard. And we agreed the minimum scope for the standard, and that was proposed and elevated to BSI for further treatments to see whether it will be developed into a standard or not. We don't know the answer to that. It's the vagaries of their funding,
Yeah.
which I believe depends on Innovate UK, maybe. I don't know exactly how they are funded. But the good news is that we were able, in just a few hours of work, with the right mindset, to come up with a scope. And the scope, as you can imagine, was not rocket science. The barcode should be relatively small, easy to read, that is, to sequence. It should have the batch number, the lab of origin, some details about the organism, and a few other things I don't remember right now. So that's what it was scoped as the minimum common denominator. Of course, it's not the most exciting science from a point of view of science, not the most exciting. But actually, if we could come together as a community, and when I say community, I don't mean only academics, I mean as a UK PLC, universities, regulators, uh, standard institutes, metrology, etc., industries, to devise and establish a standard, then things can move much faster. Uh, but right now, the weakness is that there is no standards. Lots of people agree that will be very helpful for many, many reasons, but still we haven't agreed on a standard for that.
Okay. Yeah, that makes sense. Yasir, would you like
So,
to?
yeah, I will give some simple answer to what can be limitation. Like first of all, I think that one limitation is the selection of precise site. For example, if we insert the barcode some random place, the computational prediction is not that much precise, and we insert barcode somewhere in the important regions, obviously it will disrupt the microbe and all its function. So one is this limitation. Another can be like with time over many generation, how the stability and how the barcodes can propagate through many generation when the growth or increases of the microbes. And also the barcode should be unique and easy to read. But in complex environment, I think with many organisms this can be challenging. And finally, you can say that if we have many microbes, that can be also time consuming to synthesize the barcodes, obviously. If we need unique barcodes for each
Yeah.
strain.
Okay. Perfect. Thank you. So, you recently presented at our early career researcher conference, which was Engineering Biology for Environmental Sustainability. It was so good to learn more about your work. It was a brilliant presentation. Thank you. Um, it was really good, I say, to learn more about what you're doing. You're working around identifying neutral sites for synthetic barcode integration. Do you mind telling us a little bit more about your work, whatever it is you can tell us about what you're up to at the moment?
Yes, and for the conference, yes, it was very amazing conference. And yes, currently I'm still working on the neutral sites as presented in the conference. Like we have three approaches for neutral sites with a bunch of filters, a kind of subtractive genomics for neutral sites. Like subtracting first to be a genome, then we apply one filter, then second, then third filter with kind of subtractive genomics to select the candidates neutral sites. Beside this, I am now working on developing pipeline a complete package which will predict nucleosides using all approaches. And we will collaborate with experimentalists from GitLife to optimize the pipeline by doing the experiments, which we predict are actually works or not. And finally, I'm also working on the biosecurity side. So I'm working on the sequence screening, which actually check the synthetic DNAs. Either it can be harmful or safe to synthesize those sequences. And finally, we also aim to what we are doing conventionally to simple pipelines. We're aim to develop also AI models for these. When we have enough data and we think that now we can develop AI models, then we will aim to develop the AI models for our conventional pipelines, which will speed up even more the prediction and hopefully accuracy too.
Fantastic. That sounds brilliant. Thank you. So, Natalio, your work it spans obviously things like machine intelligence, complex systems, synthetic biology. Look into the future. How do you envision the evolution of barcoding, particularly within the field of engineering biology?
Well, that's difficult
Yeah.
to say because uh, one could speculate some very fancy kinds of barcodes, and watermarks and all of this. But I think, quite frankly, I think what we really need in the next 2 years, 3 years, 5 years maximum, I would say, are to come up with a bare minimum base standard for what information a barcode should capture, and then start thinking seriously about how we can expand this standard for a basic barcode, generic barcode, into application specific barcodes. For example, it might be the case that if you want to introduce a barcode in a strain that is used for in a yogurt line or something
Yeah.
like that, you need some kind of information there that is not the same as if you will use barcodes for organisms that are used to produce some chemical products in a 10,000
Mm.
liter bioreactor, or if it was a barcode to be used in a microbe that is, for example, potentially going to be released in a river to clean heavy metals. So I think uh, we need first to establish what is the absolute minimum that we can all work together and agree on. And then we really need research to say, okay, for this kind of application, what is the next level of sophistication we need? What are the molecular biology tools that you will need to be able to do this barcoding and reading very, very easy within that application? And then also have the informatic infrastructure to then tie all of this up. So then the public, the regulator, a customer that wants to buy one of these things can all have access to this data. So I think this, from my perspective, might not sound very exciting, but I think it's really hard to do. And but at the same time, I think it probably will have more impact, more consequences, potential consequences.
I think it sounds great. I think it sounds exciting. So this whole topic has been so interesting for me. I've really enjoyed learning more about it as well. Before we finish, I do have some fun little questions for you. Don't you have to take them too seriously or anything like that. So feel free to answer how you see fit. The first one, if you could hide a song in a barcode, which track would you pick and why? And this is for both of you, if you want to.
Do you want to go first, Yasir? You have any idea?
Yes, I recently listened to a song, Back to Friends. It's a recent song just released in 2025. So I think it's related to the friends, Back to Friends. So like teenagers also were tiny friends
Yeah.
in the environment.
I like that.
So it's like giving each microbe a secret friendship anthem, which we can store inside the barcode.
Nice.
I love
So
that.
I would go for Back to Friends. It's a
Back to
nice
Friends.
Yeah.
That's a really good one. You've got to follow that now.
Yeah. No, I cannot top that up, but I thought about Katie Meloa's song called What It Says on the Tin. Someone say, what is this on the tin? And it's exactly what the barcode is for,
Yeah.
to tell you what this is about, right? We result with to decode the genome and all the effort that goes with it.
Yeah. I love that. Those are really good, actually. They're really good. I was trying to think as well. I was like, I have no idea. So, okay, let's set the scene. You're at a house party in the kitchen. Everyone's always in the kitchen. What is your go-to party fact about engineering biology that always surprises people? If you're trying to explain what you do, how do you say it?
Okay. I will stretch the concept of engineering a little bit.
Yeah,
But I will just simply tell them that actually we've been doing engineering biology for approximately 30,000 years. It started with the domestication of the cats around that time. Sorry, of the dog around 30,000 years ago. We have domesticated so many other animals and plants. And it's engineering biology without knowing engineering principles and without biology principles. So now we actually know what we are doing.
Yeah.
And this is why I'm less scared of things going wrong now than scared of how we got here without any major any major issues. Yeah.
Perfect. How about you, Yasir? What's your party fact?
Actually, I will discuss about the engineering biology concept, which actually also surprised me when I knew about that concept. The genetic circuits. Actually, genetic circuits are the genes or dots connecting to introduce entirely new pathways into the microbes, basically giving them brand new abilities they never had before. So they modify the microbes, their pathways, and giving them new function. And when I knew about the gene circuits, and that actually also surprised me in the past for the first time I hear about the gene circuits. So I think yes, people were surprised when they know about the gene circuits, especially because we know the electric circuits.
Yeah.
But that's the term, the gene circuits.
Perfect. Thank you. So, right, last question for you both. So if funding and technical limitations, they weren't an issue, so you can go as big as you want with this, what would you want to try next?
Jesse, you
Yes, my answer is very straight and simple. What I want to test lot of neutral sites
Okay.
because currently we only test a few because of we have to manage the resources, not to waste the resources.
Yeah.
So if we have unlimited resources, I will try to test a lot of neutral sites so we can optimize our pipeline, which we are doing. Currently, if I perform analysis, for example, if I got, for example, 50 neutral sites, so we will test some. We cannot test all the 50. So I am thinking if I have a budget unlimited resources, I will try one each neutral site to test each neutral site. So it will give the broad picture of the microbe where the actual neutral sites are.
That's great. How about you, Nat? What would you do?
I think I don't know. I think that Yasser is very good and very pragmatic. Actually, so probably I will vote for his idea, not mine. I think I would like to barcode the Inococcus radiodurans, send it to the space station, whether the International Space Station or the Chinese Space Station, paint the exterior wall of the space station with the barcoded the Inococcus radiodurans, leave it there for a few years, and then go and collect some samples a few years later and see what happened with those barcodes. Are still they there? How mutated and something like that. I don't know.
Stuff
Yeah.
stuff is yeah, but it's go big or go home, isn't it? You're going to space. Perfect. Thank you. It's brilliant, honestly. Thank you both so much for your time today. And I've really, really enjoyed learning more about your work and everything you're doing. I'm very excited also to see where the research and the work within EBIC and elsewhere takes us. So thank you both very much for your time.
Thank you. Thank you for inviting me. It was fun.
Thank you. If you haven't already, please do take a look at our social channels. We're now also on Blue Sky under EBIC Hub, and we are also very excited to announce that we'll be launching a webinar series very soon. If you head over to our website, ebicentre.co.uk, that's ebicentre.co.uk, you can see all the details are on there, and also a chance to register early interest. We would love to hear from you as well. If there's a topic you find fascinating or something you want us to discuss on the podcast, then do get in touch. But that's all for now, and thank you so much for listening.